Integrative omics analysis represents a powerful and increasingly crucial approach in modern biology. It aims to understand complex biological systems by combining and analyzing data from multiple omics disciplines. This method offers a comprehensive perspective. This approach moves beyond the limitations of studying biological components in isolation. It seeks to unravel the intricate networks and interactions that govern life processes. The integration provides a more complete picture. Examples of omics disciplines include genomics, which studies an organism's entire genetic material. Transcriptomics focuses on the complete set of RNA transcripts. Proteomics investigates the full complement of proteins. Metabolomics examines the complete set of metabolites within a biological sample. Other related fields, such as epigenomics and microbiomics, also contribute to this expansive view. Each of these disciplines provides a unique perspective on biological function. Genomics reveals the blueprint of life, while transcriptomics shows which genes are actively expressed. Proteomics indicates the functional machinery of the cell. Metabolomics reflects the metabolic state and cellular activity. While each omics discipline offers valuable insights, its perspective is inherently limited. A single omics approach cannot fully capture the dynamic complexity of biological systems. It provides only a snapshot. The true power emerges when these diverse datasets are integrated. This integration offers a more comprehensive and nuanced understanding than any single omics approach could provide. It allows for a holistic view. This holistic understanding is superior because it reveals connections and dependencies across different biological layers. It helps to bridge the gap between genetic information and observable phenotypes. This is essential for advanced biological research. The core principle of integrative omics analysis is to move beyond individual datasets. It aims to synthesize information from various molecular levels. This synthesis reveals a deeper understanding of biological processes. This approach identifies relationships, patterns, and causal links that are not apparent when data are analyzed in isolation. It connects different biological levels, from genes to proteins to metabolites. This creates a multidimensional view. Consider the integration of genomics and transcriptomics. This combination reveals how genetic variations, such as single nucleotide polymorphisms, influence gene expression levels. It links genotype to phenotype at a fundamental level. Transcriptomics combined with proteomics illuminates the translation of messenger RNA into functional proteins. This integration helps to understand post-transcriptional and post-translational regulatory mechanisms. It clarifies the flow of genetic information. Further integration with metabolomics allows researchers to understand how these molecular interactions impact cellular function and overall phenotype. It connects the molecular machinery to the metabolic output of a cell. This provides a functional context. For instance, a genetic mutation identified through genomics might alter gene expression, as seen in transcriptomics. This altered expression could then lead to changes in protein abundance or function, detectable by proteomics. These protein changes, in turn, could affect metabolic pathways, which are observable through metabolomics. This integrated view traces a biological event from its genetic origin to its functional consequence. It provides a complete narrative. The mechanism involves sophisticated computational and statistical methods. These methods are designed to find correlations, build predictive models, and identify key regulatory nodes within complex biological networks. It is a data-driven discovery process. By connecting these disparate data types, integrative omics analysis constructs a more complete model of biological systems. This model is crucial for understanding health and disease. It moves beyond reductionist views. The integrated approach offers numerous advantages in biological and biomedical research. It enables the identification of novel biomarkers for disease. These biomarkers are crucial for early diagnosis and prognosis. It facilitates the development of more effective therapeutic strategies. By understanding the molecular underpinnings of disease, targeted interventions can be designed. This leads to personalized medicine. Integrative omics provides a deeper understanding of complex biological phenomena. This includes processes like aging, where multiple molecular changes occur simultaneously. It clarifies the multifactorial nature of these processes. It also sheds light on disease progression, revealing the molecular events that drive pathology over time. Furthermore, it helps to understand environmental responses, showing how organisms adapt to external stimuli. This is vital for ecological studies. In cancer research, integrative omics has specific and profound applications. It helps identify driver mutations that initiate and promote tumor growth. This knowledge is critical for developing targeted therapies. It also predicts drug response by analyzing the molecular profile of a patient's tumor. This allows clinicians to select the most effective treatment, minimizing adverse effects. It moves towards precision oncology. Ultimately, this approach personalizes treatment plans for cancer patients. By integrating genomic, transcriptomic, and proteomic data, clinicians can tailor therapies to an individual's unique tumor characteristics. This improves patient outcomes. Beyond medicine, integrative omics has significant applications in agriculture. It helps to improve crop yields by identifying genes and pathways associated with desirable traits. This enhances food security. It also enhances the resilience of crops to environmental stresses, such as drought, salinity, and pests. By understanding the molecular responses to these stresses, scientists can engineer more robust plant varieties. This supports sustainable agriculture. The ability to integrate diverse data types allows for a comprehensive understanding of plant biology. This leads to more efficient breeding programs and improved agricultural practices. It addresses global challenges. Despite its immense potential, significant challenges exist in integrative omics analysis. One primary hurdle is the sheer volume and complexity of the data generated. These datasets are often massive and heterogeneous. Managing, storing, and processing petabytes of omics data requires substantial computational resources. The diverse formats and inherent noise within each omics layer add to this complexity. This necessitates robust infrastructure. This field requires sophisticated computational tools and algorithms for effective analysis. Standard statistical methods are often insufficient for handling high-dimensional, multimodal data. Specialized bioinformatics pipelines are essential. Furthermore, integrative omics demands expertise in bioinformatics, statistics, and the underlying biology. A multidisciplinary team is often necessary to navigate the complexities of data integration and interpretation. This requires specialized training. Data integration considerations are critical. Careful consideration of data quality is paramount, as errors in one omics layer can propagate and compromise the entire analysis. Rigorous quality control is indispensable. There is a need for proper normalization across different datasets to ensure comparability. Different platforms and experimental conditions can introduce systematic biases. These biases must be corrected. Harmonization across different omics platforms is also crucial. This involves standardizing data formats, units, and experimental protocols. Without harmonization, combining data effectively becomes extremely difficult. Another significant challenge lies in interpreting the results. Drawing meaningful biological conclusions from integrated omics data is often difficult. The sheer number of potential interactions can be overwhelming. This requires a deep understanding of the underlying biology of the system being studied. Without biological context, statistical correlations can be misinterpreted or lead to spurious findings. Biological knowledge guides interpretation. The ability to identify causal relationships within complex interaction networks remains a major challenge. Most integrative analyses reveal correlations, but establishing causality requires further experimental validation. This is a critical distinction. The field of integrative omics analysis is rapidly evolving. Continuous advancements in data analysis techniques are enhancing our capabilities. New statistical models and machine learning approaches are constantly being developed. Progress in machine learning algorithms, particularly deep learning, is revolutionizing how we extract insights from complex omics data. These algorithms can identify subtle patterns and make predictions with increasing accuracy. This improves predictive power. Improvements in computational infrastructure, including cloud computing and high-performance computing, are making large-scale integrative analyses more accessible. This democratizes the field for researchers worldwide. These advancements are constantly enhancing our ability to integrate and analyze complex data sets. The tools are becoming more powerful and user-friendly. This accelerates discovery. The decreasing cost of omics technologies is also a significant driver of progress. Sequencing, proteomics, and metabolomics are becoming more affordable. This allows for larger and more diverse studies. The increasing availability of large-scale datasets, often publicly shared, provides rich resources for integrative analysis. These datasets enable meta-analyses and the discovery of generalizable biological principles. This fosters collaborative research. Integrative omics is poised to revolutionize our understanding of biology. It will continue to unravel the intricate mechanisms of life, from fundamental cellular processes to complex organismal behaviors. This will transform biological sciences. It is expected to drive significant advancements in medicine, leading to more precise diagnostics and personalized therapies. This will improve human health outcomes globally. Furthermore, it will contribute to agriculture, enabling the development of more resilient and productive crops. This addresses global food security challenges. The future of integrative omics lies in its continued expansion, refinement, and application across diverse scientific disciplines. It represents a paradigm shift towards a truly holistic understanding of biological systems.